I'd like to share some ideas about targeting a very tough issue of drug-resistant tuberculosis by a different approach than we usually do. This is, of course, to illustrate the difficulties of bringing a potential product finally to the market. This is about preventive but potentially also therapeutic immunomodulant agents. This is the place where I come from, and we are celebrating our 400-year anniversary. This year we are proud to be among the top seed of the universities around the world. And this is a novel design of our TB department, which we are very proud of. We opened it last year, and it's built in a nice rehab orientation some few kilometers away from the main building, where we run a 30-bed TB unit in a country where we have very little TB left. Actually, we are talking about elimination of tuberculosis. Still, we believe we are part of a global village, and the cases that come to us are actually all foreign-born, and maybe they are asylum seekers or otherwise living and working in the Netherlands and hunted by this disease. This is a photographic impression of our unit that we are very proud of. Of course, we always have beautiful weather like in Valencia. And this is just to show that some of our patients do not like single rooms, but they like to be together. We have wonderful isolation possibilities with enter rooms that have low pressure and air treatment so that we do not spread multi-resistant bugs around the place. And this is the impression of our opening, and our patients are typically young as around the world. We'll see different forms of tuberculosis, of course, and many challenges. This could be active TB, but it could just as well be a paradoxical reaction in the patient that has maybe been cured for tuberculosis, and all you find here is maybe dead bacilli. You can't tell from face view. Now, my interest is in drug-resistant TB, and we have wonderful outcome results, but many of our patients are challenged with problems with their food ingestion and otherwise. So not only the drug-resistant tuberculosis patients, but also those who have a regular susceptibility pattern may be, hunted by prolonged treatment duration. And this is just an illustration of the work that we are doing, trying to apply this PK work to try optimize our drug treatment in low and middle income countries by using these dried blood spots and try to measure drug concentrations in the bloodstream. And this is some of our results that we try to study the regular drugs in drug-resistant TB. And this is most of what we are doing actually at this point in time. But we want to move forward to really target the problem of this drug-resistant TB by trying to impact on the epidemic. And the epidemic is not moving anywhere to where we like to see it happen. And this is what the problem. We have, of course, tuberculosis patients that spread their disease by inhalation. Our macrophages finally pick up the bug and process it in a not entirely effective way. We have CD8 cells trying to maybe kill those dendritic cells and other immunocompetent macrophages, be it professional or non-professional. We have different responses that we can measure. We have CD4 cells that are extremely important for our protection, but in the end of the day, we may end up with either latent or overt, apparent, active tuberculosis. Now, this looks like a granuloma where you have maybe a balance in terra, where the bug is hiding inside. But this is basically what we believe is the driving force of the epidemic. We have maybe 1/3 of the world population harvesting latent tuberculosis. And if anything happens, they may flip-flop into active disease. Now, the epidemic is not moving in the right direction. It's plateauing. And I think this is because maybe drug-resistant TB is an epidemic that is and haunting new epidemic within the TB epidemic itself. And this is a very big problem in our minds. And this is just to illustrate a paper which was published a few weeks ago in the Lancet, showing that young people may just die from potentially be treatable and potentially preventable infectious disease. And these people are just 30s, in their 30s, not old people like me and maybe some of the audience. And this is the paucity of the new drugs that we have. We have just seen this new kid on the block, betaquilin and the laminate, which is not allowed on any market around the world, but these are at least the two new kids on the block. And this drug may potentially impact on the phenotype that I'm going to discuss with you, which is the sort of low replicating, slowly replicating, not very active form of the bacillus. And the other drugs are maybe old ones that are used for the tuberculosis, or maybe drugs that are potentially toxic. Some of these derivatives may be very helpful in the future, but the other ones are old news. And clofazimine is a 40-year-old leprosy drug that we are trying to rediscover and use for, indeed, latent or persistent forms of tubercle bacilli. What about BCG? Well, we heard about BCG this morning, and it needs to be improved because it's been there for more than 90 years and is not impacting on the epidemic at all. And it may be effective in some regions of the world, like here in Greenland, where there is a recent a nice paper showing that there is indeed some protection. But in general, it hasn't changed tuberculosis around the world, as we know. And what about the pipeline of new TB vaccines? Well, there's maybe over 40 of them, and I don't suppose that you can read this from the back, but anyway, some of these components may be interesting, even for clinicians like me that want to treat tuberculosis. Here's the difficulty. Where would you like to attack? In a young child, before it's being infected in the first place, before it gets into the latent tuberculosis infection, or would you like to attack here at this point where maybe progression to overwhelming infection occurs or reactivated disease? All of these agents may attack on any one of these points. So it's very important, if you want to study it, how would you design your trial? I've been interested in this product. I've been reading about this one, and there's one that is missing out here, but it's MVACE, which I'm briefly going to discuss with you in the next few minutes. Here again, there's exposure, and maybe you want to have a preventive infection, a preventive vaccine that blocks here the infection entirely, or maybe you want to block it after infection has occurred, but before progressive infection is going to be established, or at any of the other points that you may imagine. The problem here is that we don't even have measures, surrogate measures of protection for any of the products that we are currently see in the pipeline. So the bottom line here is that no new TB preventive drug, no new vaccine has made it to the market. This was a very sad story about this novel product where 85A was inserted in a vector that did not protect children against tuberculosis infection in South Africa, as you may know. So there's not much good news yet. Still, we are interested in trying to target patients that now need typically more than 20 months of toxic and difficult to accept treatment, keeping them out of their office and out of their daily lives in their midlives, and they are haunted by this disease, which is called multidrug-resistant TB, which is typically where we miss the two major components of the treatment that makes the treatment potentially short. What we call short is 6 months of treatment still, but 2 years is something different again. So what is the primary problem of fighting tuberculosis, either by drugs or by immuno-therapeutic agents? That is, I think, this flip-flop phenomenon where the bug harvests a DOS regulon, a genetic code which makes it flip into a metabolically inactive, slowly replicating phenotype. And that's what we call persisters, or some people may consider it the dormant bacilli, which is associated with the phenotype of a latent infection, and this is maybe during drug treatment. Because why would you require 6 months or 20 months of treatment if you can just kill quickly most of the fastly replicating organisms? That is because this persister population is hardly affected by the current drugs that we have. And there's a lot of genes involved in this difficult process, and it's much more complicated than I can imagine. This is some of the work that was recently published to show how difficult it is, much too difficult for me as a clinician to understand, to be honest. There's a lot of evidence out there that this is a major focus of research, this flip-flop phenomenon that I simply call switching into the latent or dormant or persister mode. This is the idea that we had. Try to rapidly kill most of the rapidly dividing organisms, the metabolically active population, and then attack the resilient, the persister population by maybe specific drugs and specific immuno attacks, by eliciting immune responses to the phenotype that is the cause of the currently long duration therapy. We systematically reviewed the literature with the group and that we come up with these 5 candidates. Mycobacterium vaccae, the Ruti vaccine currently produced by Archivel in Barcelona, and then the Indicus preni, which was recently published about in the New England Journal of Medicine, and this agent that I'm not going to discuss for the sake of time at this point in time. And so these are the summaries of the Mycobacterium vaccae vaccine candidate, and actually the bottom line is it's fairly safe and fairly effective, but not widely used. Why is that? Because probably there's still this feeling that eliciting additional immune responses during active tuberculosis disease may be dangerous. We all know about the Koch's, Robert Koch's idea that he tried to use this immunotherapy with the old tuberculin, and it was a disaster. That is still preventing us from thinking about this potential treatment option. We're not going to go back to the days of Robert Koch, of course, but we might try to smart out the bug by first reducing the number of bacilli and then bringing this sort of treatment in addition to the conventional treatment. So this is the bottom line idea. This is a recent paper on the phase 2 trial of the Ruti vaccine. Ruti vaccine is a Mycobacterium tuberculosis strain that was cultured under stress conditions, expressing more of the, if you like, persister antigen repertoire than the usual Mycobacterium. And then it is killed, so it's not like BCG, a live attenuated vaccine, but it is a killed whole cell vaccine. This was their design, to give 3 rounds of treatment in 3 different dosages. And the bottom line is that it was, yes, it was safe, and yes, it was immunogenic, if you like. This is in the HIV positive population. where we saw enough immunogenicity on a range of different M tuberculosis antigens that were considered relevant. PPD, of course, a large array of different antigens, and the others, as you can see here. This was in the HIV negative group, and I'm just pointing at the 25 microgram of Ruti, because this is the one that they're going to pursue further. The bad news is not on the slide. The bad news was that actually there was a lot of not painful, but indurated skin at the site of infection, at the site of injection of the Ruti vaccine. It was a subcutaneous shot, but most of the study participants experienced this. So this is in healthy volunteers, but they're all latently infected with tuberculosis, and some of them have HIV, but they're all above 350 CD4 cells per microliter. So they were all in the safe range, but yes, they were co-infected with HIV. So they're believed to be latently infected with tuberculosis. So this is a proof of principle, if you like, a safety study to show that yes, there's immunogenicity, and also yes, there's enough safety to pursue. So this is the WHO readout, if you like, where there's enough a reasonable response rate on the 25 microgram of Ruti, both in HIV negative and HIV positive study participants. This was the news, of course, of 2 weeks ago, where the MIP, the Mycobacterium indicus praenae, was tested in a funny way, in my opinion. It was with the endpoint of a clinical endpoint of pericardial tuberculosis. Now, pericardial tuberculosis is, of course, a sign of tuberculosis, of disseminated tuberculosis, but this was studied in many centers around the African continent. And the bottom line is that it did not add anything good for these patients on the endpoint of pericardial disease. Now, this is a funny endpoint because I would be interested in the tuberculosis disease in general. Would it alter their cure rates, their chances of getting rid of tuberculosis? Not necessarily whether the pericardial restriction was better or worse or equal. Now, in this endpoint, there's no difference. And it was combined with prednisolone, where on the one hand you would elicit immune response, immune enhancement, the other way you would reduce the immune response by prednisolone. So I think overall it was a funny sort of experiment. But the good news about it, I would say, is that yes, it is safe to use immuno-therapeutic agents in active tuberculosis disease. And that is, I think, the good news about it. We should pursue this idea further. So this is what we plan to do. We try to collaborate with several high-burdened countries in Eastern Europe with MDRTB, and we want to try bring these advanced products to the phase 2 and 3 trials. We are, of course, looking forward to have sponsors on board to make it happen. I'm sure we'll get there with the enthusiastic team that we have. Thank you very much, Mr. Chairman and others.
Thank you too for the nice presentation. So really happy to know about the therapeutic vaccine for the anti-drug-resistant tuberculosis. So we have time for one question. Yeah.
Thank you. About the duration of treatment with TPA vaccine.
I'm not sure I get the question right.
How to use TP vaccine?
Well, this is, this may be important to. For the Ruti vaccine, it was tested only in latently infected tuberculosis cases, and now we are moving to the next stage. We want to also administer it subcutaneously. For the Mycobacterium vaccae, it is an oral product, so that is even easier for recipients, and But for the routine vaccine, there's no formulation as an oral administration, no.
What do you think about the use of immunomodulatory cytokines in lungs to try to modulate the infection? What do you think the use?
What do I think about it?
Yes.
Oh, I think a whole lot about it, but the bottom line is, of course, you want to try to elicit other intermediate surrogate endpoints, right? I think the bottom line is, I think if I listen closely to people like Tom Ottenhoff and Stefan Kaufmann in Berlin, it's general belief that we do not have robust surrogate endpoints at all. We don't know what to look for. So I think the bottom line is we have a lot of models. We should show immunogenicity and safety, but then finally go on to study ultimately in humans, in patients with TB disease, and try to see whether we can either reduce recurrent disease, relapse, or enhance the rate of sputum conversion, that is, render them sputum negative, culture negative, or any of the other endpoints, finally also reduction in mortality. I think all the other endpoints need to be validated from studying these potential surrogates and looking at the final answer. Are we going to see patients being cured in a higher percentage? Because MDRTB now kills 1 in 2 patients. 1 in 2 is actually dying from TB. That's what happened around the world.